morning, everyone. I'm very happy to be part of this Microbiology Consortium webinar series of the Philippine Society for Microbiology, a society which is very dear to me. Well, after the talk of Dr. Rodney Perez, emphasizing on the enhancement of nisin production using molecular biology techniques, I'd like to continue uh, sharing with you on again another bacteriocin, that is Pediocin, uh, talking about the upstream and downstream optimization process. So the outline of my talk, first I would like to give you a review on the Philippine fermented foods and about the lactic acid bacteria that I've been talking about, which produces Pediocin, and of course the main topic on the challenges of Pediocin production and how it can be overcome using optimization uh, methods, and then later on the future prospects, which would be the applications and future possible studies on uh, bacteriocins of Pediococcus acidolactis. Okay, so well, we all know that our country is also very rich in fermented food products, and you can find it anywhere. And basically, the northern Luzon, central Luzon area, they would prefer the meat-based or fish-based products, and in the middle part, southern Luzon would prefer the starchy or vegetable-based products. And of course, in the Visayas, they have got a lot of fresh fish and seashells, and so sometimes they would preserve them. Also, fruits. And in the Mindanao area, there are areas where they produce cassava, and they also produce some fermented sourdough from cassava, and also uh, some puto and many others. Well, these are very interesting to study with. And in the past years, we were trying to isolate bacteriocin producing lactic acid bacteria. And among them were the pedococci. And in here, you will see a list of around 13 bacteriocin producer isolate of Pediococcus. And they all basically are coming from fermented rice with a mixture of fish, also fermented meat, pork, or sausages. And most of them have the pediocin gene, the PAP-A gene. Some have the smaller sizes, but many have good bacteriocin activity against Listeria, Staphylococci, and Enterococci, and other indicator organisms. So what about Pedicoccus acidolactis? Again, as a review, we know that there are lactic acid bacteria or lab that are often found in pairs or tetrads, and they're found in various fermented food products that are quite ubiquitous. Ubiquitous, see the in the dairy, meat, sourdough, vegetables, fruit juices, silages, and others. And also, they grow in a wide range of pH, osmotic pressure, temperature. They have good thermostability, viable up to 65 degrees centigrade. And many are bacteriocin producers, namely Pediocin. Here you will see a PCR result wherein a PAP-A gene uh, are analyzed. No? They're one of the genes responsible for bacteriocin activity. Now, Pediocin, well, basically, they are class 2A bacteriocin, those with the wide G and GF motif in their N terminal. And they're potentially useful for food preservation, vet, medical, and various other purposes. In here, I show to you the purification protocol of the Pediocin from Pedicoccus acidolactici uh, to be able to determine the molecular weight by multi-top MS. It should be uh, highly purified using column chromatography and express. This is an example of uh, activity assay using a spot on loan assay. So there are several bacteriocins from lactic acid bacteria, and this has been tackled previously by Rodney, specifically the nisin and tyrosine, and those belong to the class 1 bacteriocin, or the modified one. They are smaller bacteriocin, and Pediocin belongs to class 2 or the unmodified bacteriocin, and both of them, both classes are very heat stable with less than 10 kilodalton molecular weight. And the other category, class 3, is thermolabile, and it's quite big. And the activity is not through lysis. Sometimes they are referred as bacterial license. Well, Pediocin, according to Pagiani and Anastasia, though, are class 2A bacteriocin with strong anti-listeria activity. And they share, yeah, as I mentioned before, highly conserved hydrophilic uncharged N-terminal YG and GAD amino acid sequence. The differences among the Pediocin-like bacteriocin are on the C-terminal side. Now, they're quite thermostable. Why? Because they have a couple of disulfide linkages between cysteines, as you'll see in here. And the activity is also retained at a wide pH range, around pH 2 to 9. And also, the secondary structure is random coil in aqueous and ethanol. We did an experiment on this one. I mean, a study on this one before. And also, the bacteriocin is have a leader, meaning that at first it is synthesized with 66 amino acid residues, and then later on, after post-translational modification, the leader fragment is cleaved, consisting of 18 amino acids, and uh, only the 44 amino acids, which is a biologically active peptide, remains. The estimated molecular mass is around 4.6 kilodalton. Well, exactly, it's around 4,624 daltons. There are already commercially available bacteriocin. As we all know, nisin, this is the most popular, approved in more than 50 countries. But the other types of bacteriocins are now, entering into the market, and it includes Pediocin. So the commercial name is Bactofirm FLC, being produced by Christian Hansen. Market value of bacteriocin is quite high, uh, more than half a billion no US dollars in 2020. What are the challenges of Pediocin production? Well, we all know that bacteriocin is very small, so the titer is also is quite small during fermentation. It's a high value product. Now, the producer strain is fastidious. Huh? Lactic acid bacteria. They need a good, a highly nutritious media. This is quite expensive. So we need to really optimize so that at least we can have a less costly production media. Also, downstream processing is not easy. Peptide easily adheres to various surfaces, and so it's quite difficult to use a medium with solid material. So the non-defined media, we did some experiments on this one. We tried rice bran, we tried other natural sources of substrate or carbon and nitrogen, and sometimes we don't see that much bacteriocin because it adheres to the various solid surfaces. And also there's peptide aggregation wherein the activity is lessened or it's lost during aggregation. And there are challenges of scaling it up from the uh, large scale method process, result up to the scale up. And so, how do we improve productivity through process optimization is important, and how do we optimize the downstream process is also important. Luckily, like some other pharmaceuticals, we don't need that much big fermenters to produce bacteriocins. So, how do we optimize bacteriocin? The main factors which affect bacteriocin production are the culture conditions and then the media composition. And among the culture conditions, the more important ones are temperature, agitation or aeration, right? uh, and also pH. Uh, on the other hand, carbon to nitrogen ratio is important for medium composition and other surfactants, minerals, which are needed for enhanced bacteriocin production. There are two kinds of experimental strategies in optimization. One is the OFAT method, or one factor at a time experiments, which involves testing of variables one at a time. On the other hand, the design experiments, or DOA, would allow you to experiment on many factors simultaneously. So, what are the advantages and disadvantages of these two experimental strategies? For the OFAT method, advantages is the convenience of running the experiment. We don't need to change a lot of conditions and a lot of medium composition, just changes one at a time. Now, moreover, you might just need less time to do it. However, the main disadvantages are it would require more runs for high precision and estimating effects. And more importantly, you cannot estimate the interactions among the different factors, and thus you can miss the optimal settings of the factors. On the other hand, for the design of experiments, it would cover a broader area of volume and broader area of factors, and you can draw better conclusions out of that. And it would reveal interactions among the factors, and it may require less resources because just do the runs uh, in a compressed given by the design. However, disadvantages would be the difficulty of experimenting with several factors, say more than two factors. It's quite difficult. Thus, it's important that you need to follow several steps in the design experiments. Well, this is the spot unknown method of for bacteriocin assay. Maybe a lot of you already know about it. You have the bacteriocin producer, and you have to test against the indicator organism. And first, you have to process, remove the cells, get the supernatant, and you can try centrifugation, and then also you can boil. And after that, because it's a heat thermostable, if you just we're working with class 1 or class 2 bacteriocin, and you have to neutralize the pH. And then after that, you have can do the two folds dilution so that you can estimate the activity unit, and you can do direct spotting on the indicator organism prepared from a soft agar in a two-layer, no? Double layer agar plate. And here you can see the result of a spot unknown activity assay. And a typical equation is being done depending on the amount of the spot that you'll be doing using. This is an example of the time course of growth and Pediocin production by Pediococcus acidilactici strain S3 in MRS. And the typical bacterial activity is around 6,400 AU per ml, and it would be produced during the uh, initiation, the stationary phase of growth. And also with the decrease in the pH, the post-translational modification happens. That is why it starts to produce more bacteria at a lower pH. This example, Lactobacillus sakei, was used as the indicator organism. So we did some off-fat experiments before, and we were able to optimize temperature. So one at a time, uh, we optimized some of the needed conditions. Carbon 3%, polypeptone 0.5%, calcium carbonate 1%, carbon source, sucrose give a better result comparable to MRS than glucose, fructose, or xylose. And also the yeast extract, if you add more than 2%, production is quite good, up to 3.5%. However, this would be quite costly. And there were constant variables. We added also 1% sodium 0.5% sodium chloride. The pH, you don't need to make it, control it, because you need to let it go into the acidic phase for the production of pediocin. Now, inoculation rate, this is 1%, sampling time 16 hours. And so after the off-fat optimization, we did production of 2 liter fermenter using sugarcane juice. And indeed, there was a good bacterial activity using the spot and loan method. However, uh, carbon source and yeast extract are quite high. So the nucleus rate was also 3% on here. Anyway, would that experiment be quite enough? Well, we thought that it's not that so, because it was observed that the high output was due to the higher input that uh, we gave. So we might no minimize the input with high output using process optimization. So we did also the TOA so that we can see the interactions of variables. So this is the main process of the design of experiment. As it was lectured by one of my uh, laboratory colleague, she's a chemical engineer. And anyway, the first step is to identify the process factors and responses. The factors would be the controllable input factors, uh, such as the carbon to nitrogen ratio, the yeast extract, temperature, and so on. But you will also be having the uncontrollable input factors, which would give the variation in result, errors. For example, errors in measurement, errors during sampling, and the setting of the minimum and then the maximum values for the controllable factors also. You might need also to identify the best one. So in bacteriocin production, because it's growth related, you also need to optimize biomass production. So the next step would be the screening. Well, basically, if the experimenter doesn't know which of the several factors or parameters are significant, you might be needing this step screening. It's done if you have more than 6 factors, and the significant linear effect on the response can be determined. Usually, placket Borman design is a good design for this step. And later on, follow-up experiment can be done to characterize those factors. So more information will be obtained, and then also the factors can have significant linear interaction effects on the response can be identified. It can also detect the nonlinear behavior, and you can add more center points to several type experimental designs, so you can detect possible curvature. So going into the optimum value. So examples are the factorial designs and other designs. And next would be the optimization process. Now for the optimization process, this can require several runs depending on your analysis of variance. So you can do repetitive experiments on this stage. And you have to narrow down the list of important factors, and maybe you can narrow down the coverage of, I mean, the low value and the high value of your factors. You can narrow it down to towards a more optimum region, no, or in the curvature. Now, the quadratic model can be fitted, and so second order effects now equation can be set, and also you can do the factor settings that would maximize the response or minimize, no, some values, some inputs. And lastly, confirmation is being done, confirmatory run. Usually you can use the fermenter to use the several factors. However, also in agitation and aeration, even before confirmation, you can use a fermenter or bioreactors. Now, this would ensure if your result is okay, your model would produce the accurate prediction for the responses, and then the process would be robust under field conditions, under scale-up conditions. Okay, so I will give you an example of one uh, experiments done by some of my NBB 240 students. So in here, so it's just a defined medium, meaning it's synthetic media that we use. Now for the bacteriocin assay, instead of the spot alone, we use the agar well diffusion method because it's better. It's easier to do it, but you need more. You need more amount. They use 30 microliter. Hence, the activity would be not that high if you use a 10 microliter of the spot alone. So again, bacteriocin activity would depend on the assay method that you'll be using because those are just arbitrary. Okay, now this equation in here, you can see that the n in here to n uh, now would equate to okay, so the number of wells, yeah, that you have witnessed the 2 inhibition activity. In here, maybe you can count 1, 2, 3, 4, 5, 6, 7. So 2 raised to the 7 times 1000 over 30. Now you can have the value. Also, more importantly, we use the Listeria NCO also indicator organism. It's a surrogate indicator for Listeria monocytogenes, the main target bacterial pathogen of Pediocin. Now, we set the low and high values. We did not anymore pass through the screening method because there were several literatures already, and that would at least give a hint on how much low value and high value to use, and also which would be the more significant factors. So for carbon source, we use sucrose and peptone for the nitrogen source. and also the inoculum level. So three factors were used. So these are the low value and the high value, 10, 30, for peptone 5, 20, and for inoculum zero point 0.5 and 3. And so using the design expert, uh, the software gave this one, no, the values to use based on the low and then high settings. So if you will notice, there are values that are similar in here. So maybe five of them, and in the box banking design, those are the center points. And so 17 flasks were done, were made, and then activity was estimated. Also, the absorbance or the biomass also was estimated. And the results were analyzed using ANOVA. And in here, you will see that the model use was significant. And also, the lack of fit was not significant. But among the factors, only uh, peptone was the significant factor that was identified. And second order equation was given as this one. And so from here, the predicted value can be computed. And so the activity, bacterial activity, would be computed based on the factors that were used. And also the software gave uh, three-dimensional graph or surface model, no, of the optimization result, showing the interactions of the various parameters used. So in here, uh, the interaction among sucrose and the inoculum on the bacterial activity is shown. And in here, interaction between peptone and also inoculum. In here, sucrose and peptone. And the red, orange, almost red, are the optimum values. The dark blue would be the minimum values for the bacterial activity. So below, also the graph for the predicted value based on the equation also shown in here. And there's not much difference. So that is why plotting the residuals, and you will see in here that there's not much difference among the normal values and versus the residual value. Uh, Fit is very good, and the R squared in here you will see is quite high also, almost 0.87. So the next step would be the numerical optimization, meaning the software would advise you what would be the optimum values that would give the bacterial activity, which is very desirable. So in here, 71 models were generated, and the maximum desirability value was 0.865. And from these optimum components, a good bacterial activity was obtained, can be obtained. No? Uh, these are predicted values, so it needs to be confirmed. So we did confirmatory bioreactor runs using the optimum parameters, and interestingly, the bacterial activity increased. And more importantly, only after 8 hours, a good bacterial activity was obtained. 8 hours. So it will entail savings in the fermentation time and resources, and also savings on the input. Uh, so that was good result on the optimization. And what about recovery and purification? So depending on the use of bacteriocin, or for some pharmaceutical or other um, delicate uses, we need to purify them. Good purification is necessary. However, for other uses, just mm, simple purification would be enough. Now, as seen in the literature, most of the purifications done were uh, on chromatography after precipitation and then HPLC, so that it can be fully characterized. And we also did work on that, and we just we did not do any chromatography, but we just did the adsorption desorption method, adsorption at a low neutral pH, and then desorption at a low pH. So the method was quite simple enough, and to produce a bacteriocin-rich mixture. Now, after RPH PLC, the yield was quite good. It's 40.4%. So this result, we tried to think of how would we use other materials to further increase it. And so it was suggested, no? Uh, Dr. Migo, Dr. Rofara, the student, their student, tried to use a nanozeolite. And nanozeolite powder was used as the adsorbent together with the dead cells for the absorption. And this is the mechanism uh, wherein the protein would be absorbed on the pores, removing, no, the ions, cut ions, and it can be desorbed by adding anions. So this is a simple way of adsorption desorption protocol. That is why for the experiment, we just, for the absorption experiment, only two factors were used. The nanozeolite concentration set at 5 for a low value and 15 for a high value, and the contact time set at 10 and 14. These were based on previous literatures. Then for the desorption experiments, the sodium chloride was used to desorb it, and the low value was zero point 0.5, high value is 1.5. And also another factor was the time for desorption. The low value is 30, high value is 90. So these are the results. The ANOVA showed that the model was significant, very good uh, result, and lack of fit was not significant. Uh, and more significant one was the nanozeolite concentration in here, but not the time. No? So this is the three-dimensional graph and the polynomial equation obtained to predict the value. And during numerical optimization, desirability was quite good, 0.931, and the optimum nanozeolite concentration uh, from the equation was obtained as 18.16 gram per liter, contact time of less than 10 hours. What about the desorption? For the desorption, also the model was good. It was significant, while the lack of heat was not significant. And desorption time was the one that's significant, but not the sodium chloride concentration. And the equation, from the equation, the optimum conditions were estimated to be around 1 molar for sodium chloride, with a desorption time of um, more or less 1 hour. So as a summary, the actual value based on the experiment of the activity, but this is just estimated based on the zone of inhibition. was very in good agreement with the predicted one, and the purification also was good in a good agreement. And moreover, uh, the method using nanozeolite sodium chloride absorption desorption method had a somewhat higher recovery. It was improved compared to our previous method of pH cell biomass absorption desorption method, and purification factor also improved. Hence, with this, it can now be tested, I guess, in a larger scale later on. So that would include the future prospect. And moreover, there would be a development of products for biomass. Uh, we can use them in various applications. Also for the bacteriocin, you can just use the bacteriocin, the crude bacteriocin, in trying to inhibit some products, the bacterial pathogen like enterococci or listeria in white cheese, and also some drink. Uh, Another one is anti-helicobacter pylori. P. acidilactis was found to be quite effective in inhibiting the intestinal or stomach absorption of Helicobacter pylori. And also the bacteriogenic uh, acidilactis can also be used as probiotics in a fish, feed supplement, also in animal, also in xylem can be used. And moreover, the Pediocin can be used in stainless steel services. Some good results were obtained in here using a hard drive technology by one of our master students. Dr. Zamora was the advisor of that research. Also, Pediocin would be used in perhaps no in wine fermentation. And most importantly, this one, the pharmaceutical use, it was in one of our experiments, it was found effective in inhibiting cancer uh, cells. So these are the possible prospects. And moreover, with the advent of postbiotics, it's not only the bactericin, but the other peptides, other metabolites, organic acids, enzymes can be fully recovered. And maybe in a particular application, it can be used. It can be less costly for downstream processing and to have good prospects in the future. So this is all for now. Thank you very much for your attention. And I would like to acknowledge several people, a lot of them, that had been in collaboration with me and with our group in doing research in bacteriocin and lactic acid bacteria. And more importantly, our students, no? Cheming students, food science students, and MEB 240 students. They were all very good. And uh, I would like to thank everyone for listening to this webinar. Thank you, and then stay safe always. God bless everyone. Mabuhay po ang lahat.